Thank you for inviting me here today. I'm going to talk about some of our work in triple-negative breast cancer, in particular a grant that was just awarded, which is our grant, the Innovative New Collaboration Grant, that was awarded to myself and to Dr. and Dolly at University of Delaware. But this grant is part of a bigger initiative at ChristianaCare that encompasses our health equity team, our population health team, as well as our team in the Poly Center. So we are focused on triple-negative breast cancer, and just a little bit of background for those of you who may not be familiar. This is a subtype of breast cancer. It's about 10 to 20% of breast cancer cases. It's called triple negative by what it lacks. It lacks 3 different receptors: estrogen, progesterone, and receptors. And the important thing about this is that we have targeted treatment to estrogen and progesterone and HER2 receptors. And so without those targeted treatments, triple negative breast cancer really doesn't have any new therapy and is treated with standard chemotherapy, and it is usually resistant. Women with this type of breast cancer develop early metastases. It is a fast-growing cancer and has worse mortality than other forms of breast cancer. Importantly, African-American women are more affected by triple-negative breast cancer, and it's in fact responsible for about 50% of the health disparity that we see in breast cancer. So while some of this is a screening disparity, we do believe that there's a biological disparity as well. So why do we care about it at ChristianaCare? Delaware is ranked #1 in the country for incidence of triple-negative. So both overall incidence of triple-negative breast cancer and the black-white disparity in incidence of triple-negative breast. So our group, in collaboration with our population health team led by Dr. Scott Siegel, has been investigating why we might have this high incidence in Delaware. And Scott has identified these hotspots highlighted here by the red circles, where there's a higher incidence of triple-negative breast cancer compared to other types of breast cancer. So not incidence of breast cancer overall, but a specific subtype of breast cancer in these hotspots. And through several different studies, we've shown that in these areas with high triple-negative breast cancer, there's also a high incidence of what we call exposure. So exposure to breast cancer risk factors, as well as environmental exposure. So and particularly looked at parity without breastfeeding, obesity, alcohol use, and pollution in these studies. And it's been shown by other groups that in areas that have these high exposures, which usually are associated to lower socioeconomic status, that there is an increase in inflammation, which we know is also a risk factor for development of triple-negative breast cancer compared to other types of. And we looked at women with breast cancer in these hotspots, and we do see that there's an increase in inflammatory cytokines in women with triple-negative breast cancer from hotspots versus women outside the hotspot. And we also see a pattern of epigenetic changes in these women, and we know that some of these exposures do induce epigenetic changes. These are particularly in the peripheral blood cells. So we're continuing to investigate these epigenetic changes, also looking at epigenetic age in these women who live in the hotspots to see if and use that to predict risk of triple-negative breast cancer. When we also look at these tumors, we can see that these tumors look very different. They are very inflamed from women from the hotspot compared to triple-negative breast cancer or estrogen receptor positive tumors that are outside the hotspots. And we are currently working to understand this. We don't know if this means that they may be more responsive to immunotherapeutics or not. So right now we are doing some spatial transcriptomic profiling to really understand the difference between the triple-negative hotspot, or women who have triple-negative and live in hotspots versus women Our hypothesis really is that there's a combination of socioeconomic factors, genetic factors, and breast cancer risk factors that contribute to this incidence of triple negative breast cancer. And we believe that all of these factors, or it's known that a lot of these factors can actually increase chronic inflammation, which we think then promotes not just your risk of breast cancer, but your risk of getting triple negative breast. So we know socioeconomic factors such as pollution, alcohol use, obesity, all increase circulating cytokine levels. There's some genetic factors, BRCA1, that is a risk factor for triple negative breast cancer that increases early inflammation in the breast tissue itself. And some other genetic factors, one of which I'll talk about in just a second, that's the focus of our grant, that increase systemic inflammation. And then breast cancer risk factors, breastfeeding or not breastfeeding can increase localized inflammation within the breast. Comorbidities such as diabetes can also induce inflammation. And so these are some of the factors that we found in our triple negative hotspots. So one of the genetic mutations that we want to look at, particularly in these hotspots, is the DE-phenyl allele. And this is a SNP that is associated with increased inflammation. It's most prevalent in women of African descent, who are also the women who have the highest incidence of triple negative breast cancer. This receptor, the DARK receptor, is a chemokine receptor that is the receptor for malaria. And it's believed that this SNP developed as a way of malarial resistance. But the role of DE-phenyl is to take up chemokines and remove them from, or the role of the DE gene is to take up chemokines and remove them from circulation. The women that have the SNP aren't able to do that, and so they have increased circulating inflammation. And it's been shown that the DE-phenyl allele is associated with increased incidence of triple negative breast cancer in women of African American ancestry. So we believe that all of these factors, DE-phenyl allele combined with socioeconomic factors and breast cancer risk factors, are contributing to this chronic inflammation in our hotspot areas and inducing increased incidence of triple negative breast cancer. So we are currently doing this study looking at women who don't have triple negative breast cancer yet, who live in these hotspots, to identify our incidence of DE-phenyl allele in these women, and also assessing their exposure rates, if there's a relationship between presence of DE-phenyl allele, neighborhood in these hotspots, and systemic inflammation. So that is, we just got the grant funded. We had planned to do some blood drives partnering with Delmarva Blood Bank, be able to recruit patients into the study. We have some issues. Delmarva is undergoing leadership change and isn't able to fill that role right now. So we're looking at using some of our mobile health vans and health teams at ChristianaCare to recruit for this study. So I'll be happy to come back maybe in a year and give you an update on any results that we may have. I'd like to thank my collaborators and all the women who have donated blood and tissue samples. And I will stop there